Okay, so I'd just like to thank Jill and Natalie for the invitation to come and talk to you all today about donating your brain for research. My name's Tamrin Lashley and I'm a research fellow currently funded by Alzheimer's Research UK to carry out my research at Queen Square Brain Bank. So I thought for this session I'd like to give you an introduction to Queen Square Brain Bank, who we are, the actual process of brain donation, how we pathologically diagnose those brains with fFTD, and then I'll give you a brief summary at the end of the current research I'm involved with and research that's been published recently. So who are we? We have a clinical director, Professor Tom Warner, who's recently taken over from Professor Andrew Lees, a renowned clinician and research scientist in Parkinson's disease. We have a recently newly appointed neuropathological director, Professor Janice Holton, who recently took over from Professor Thomas Rivez, who I personally worked with for 15 years. We have a variety of clinical research fellows at the Brain Bank, and we also work closely with the clinicians at the Dementia Research Centre. We have about 4 or 5 research scientists and postdocs carrying out their own individual research projects on frontotemporal dementia, Alzheimer's disease and multiple system atrophy. We have a selection of administrative staff who I'll introduce in a moment, and students range from PhD students who are with us for 3 to 4 years, and MSc and BSc students that carry out their postgraduate projects with us. We collect brains from movement disorders like Parkinson's disease, all types of dementias, of course normal controls, which we're always in need of. So our administrative staff are Linda Parsons and Lynn Haddon, so anybody interested in donating their brain, these 2 people are the first points of call, Linda and Lynn. We have 2 senior research technicians, Kate and Rob, who deal with the brain donations when they arrive at the Brain Bank, and then a Brain Bank nurse and PAs to the neuropathologist and clinical director, and the scientific research fellows that carry out the research projects within the Brain Bank, and our support staff, the research technicians in the lab. So I thought I'd talk you through the process of brain donation, and the most difficult decision obviously is to decide whether you want to donate your brain for research. But I thought I'd highlight the process and how we deal with the brains at the Brain Bank, which may give you a better indication of how your brain will be dealt with and may help your decision be more informed of that decision in the future. So the paperwork needs to be completed at Queen Square Brain Bank, and then prospective donors are placed onto a register within the Brain Bank. At the time of death, the next of kin contacts Queen Square Brain Bank and will contact Lynn and Linda, who I've just shown you the photos of, and they'll take them through the process of the actual brain donation. They'll arrange the autopsy and arrange for the brain to come to Queen Square Brain Bank, where it will be handled by our research staff in the lab. Once the brain's received, it's dissected and retained at QSBB at Queen Square Brain Bank for the neuropathological diagnosis. Once it's received its official diagnosis, it can then be released for research within UCL nationally and internationally for various research projects. When we receive the brain, it's hemidissected, so we split the 2 hemispheres in half. 1 half we freeze and 1 half we fix. So the frozen half is coronally sliced into these thin slices and immediately placed in a -80 freezer for research projects. The other half, usually the left hemisphere, is fixed in formalin for 3 weeks, awaiting for the neuropathological diagnosis. So once it's been fixed for 3 weeks, the fixed half also is cut coronally into thin slices. These slices are then assessed by the neuropathologist, because even at the macroscopic level we can have clear indication of what the underlying pathological process was, and I'll highlight some of this later in the talk. We then take representative regions from different brain areas that covers all the diseases that we collect at the Brain Bank, and every brain is sampled in exactly the same way. So we collect the hippocampus here, which is always block 10, so in every case everything's standardised for every disease. These blocks are then taken from the tissue and placed into a wax block. This process takes another week in a machine. It goes through various chemicals, and then we embed it into these wax blocks. Using these wax blocks, we can use a machine called a microtome to cut really thin sections of the brain and place these on 2 glass slides. We'll then use a process called immunohistochemistry to stain any abnormalities that are caused by the disease that we can then see down the microscope. So the process of immunohistochemistry, I thought I'd just outline this, seeing as I mentioned it quite a few times in the talk, is a process where we need to label what the abnormalities are in the brain. So we take the thin wax sections on the glass slides. We then need to remove the wax, revealing the abnormalities that we want to look for in the brain. So in this slide here, these 2 red circles are the abnormalities, and we need to be able to see these down the microscope. So we use various antibodies to build up different layers on these abnormal structures, which then we can tag with a colour, and then we can see these colours down the microscope to where the abnormalities are in the brain. So as you know, frontotemporal dementia can present with 3 major clinical symptoms: the behavioural variant, progressive non-fluent aphasia and semantic dementia. But non-genetic variants of these diseases present in clinic can have multiple pathological processes happening in the brain. We have other clinical symptoms associated with the disease, and the frontotemporal degeneration is known pathologically as frontotemporal lobar degeneration, or fTLD. So the pathological classification of fTLD has evolved over the last 10 years to encompass all these diseases we see at the bottom here. But back in 2006, we only could split it into 2 subtypes of fTLD. Those containing the tau abnormalities, a protein in the abnormal structures, or those contained without tau in the abnormal structures. And I thought I'd highlight some of the diseases, showing you macroscopically what we see in the different brain slices, and then what I'd see down the microscope to be able to give the diagnosis of the different diseases. And if I start with what we call tauopathies, so the abnormal protein or abnormalities we see in the brain are made of the tau protein. This can be further divided into those with 3 repeat tau. So tau can come in 6 different isoforms, and we have diseases that are split into 3 repeat tau or 4 repeat tau, or a mixture of both. So if we start with Pick's disease, this is a 3 repeat tauopathy. And when we receive the brain, we can already see abnormalities on the macroscopic surface of the brain. So we can see there's a reduction in the height of the frontal lobe here and here. And then microscopically, once we've cut the brain, made the thin slices and stained for the abnormalities, we can see these brown structures here called Pick bodies that are made up of the 3 repeat tau isoforms. And these are unique characteristics of Pick's disease, are these brown Pick bodies, small spherical structures that are on the side of neurons called Pick bodies. If we go back to the classification, and then we move to the 4 repeat tauopathies, so these are diseases that have only 4 repeat tau in their pathological abnormalities. We can look at corticobasal degeneration, and again we can see a slight reduction here in the frontal cortex, and then when we look at the slices, it's more evident that there's a reduction in the bulk of the frontal cortex here, and also an enlargement of the lateral ventricle. And then down the microscope in corticobasal degeneration, we'll see distinct structures called pretangles here, again made up of the protein tau, and then astrocytic plaques. So these are abnormalities of the tau protein that build up in the cells called astrocytes, which are glial cells that support the neurons. Another 4-repeat tauopathy is progressive supranuclear palsy. So it's the same protein that affects corticobasal degeneration, but it has characteristic hallmarks on the macroscopic slices and in the structures that we see down the microscope. So on the macroscopic slices, we can see a small structure called the subthalamic nucleus here, and this is abnormal. It's quite a dark colour here. On a normal brain, this would be an almond-shaped structure with no discoloration. So this, when we see this on the slices, it's quite indicative that this may have supranuclear palsy. We also see the superior cerebellar peduncle is smaller than it should be in a normal brain, and the dentate nucleus is often blurred and discoloured in the cerebellum. Then looking down the microscope at these diseases, again we see abnormalities in the tau protein in neurons and in glia, but this time the structure of the abnormalities in the glial cells are different. So instead of seeing the astrocytic plaques, so abnormalities of tau radiating around the glial cells, we now see tufted astrocytes, so the deposits are in the centre of the glial cells. So if we move back to the classification and move to those cases with tau-negative abnormalities, this can be further subdivided into those that have TDP43 positive inclusions and TDP43 negative inclusions. If I start with the TDP43 positive inclusions, this can then further be subdivided into 4 different subtypes. And these 4 subtypes can be associated with different clinical presentations or different genetic abnormalities, which I'll highlight later. So in TDP type A, if I just show you quickly the different abnormalities that we see, that we can then classify them into the different subtypes. So in a type A, we see small dystrophic neurites, so these little brown blobs here. And we also see cytoplasmic inclusions, similar to the PIC bodies that we saw in the PIC's disease, but now these are abnormalities made up of the TDP43 protein instead of the tau protein. And we also see deposits within the cells themselves, and we call these neuronal intranuclear inclusions. So the fFTD TDP type B is characterised by neuronal inclusions, again deposits of the TDP43 protein around the cells, but this time the shape and characteristic of the inclusions are more granular. It affects all cortical layers within the brain rather than just cortical layer 2 seen in type A. And we very rarely see the intranuclear inclusions like we did in type A. Type C and type D are probably the easiest subtypes to characterise down the microscope. In type C we have these long corkscrew-like neuritic threads here, which are quite distinctive down the microscope. And in type D, all we see are the intranuclear inclusions, so the deposits within the cell rather than any cytoplasmic or neurites as seen in the other subtypes. A recent genetic abnormality, the C9orf72 gene, is quite interesting from the pathological point of view, because it's the first time we're able to identify a genetic abnormality down the microscope. We see all cases with C9orf72 have the build-up of TDP43 in inclusions. We also have this additional pathology made up of P62 deposits, and I think Adrian will probably go into this in more detail in his talk. But this is the first time we see deposits associated with a specific genetic abnormality. For instance, if a case comes in with a known progranulin mutation, we wouldn't be able to identify that that case has a specific progranulin mutation. We'd only know that it has TDP43 deposits within the brain. But when we see these deposits down the microscope, we know that it has the C9orf72 mutation. So I've shown you that there are 4 subtypes of TDP43. And we've done large clinical pathological studies with the clinicians at the Dementia Research Centre, and we've been able to pinpoint certain clinical manifestations and genetic abnormalities with the specific subtypes. So if we see a type A subtype for fTLD TDP, it can be associated with a progranulin mutation or the C9orf72 mutation, and it's usually associated with a behavioural variant clinical manifestation. Those with associated motor neuron disease clinical manifestations are usually associated with a type B subtype, and again this can also be associated with the C9orf72 gene. A semantic dementia, if you have a clinical presentation of a semantic dementia, this is usually always a type C pathology, and a type 4, sorry, that should be D, that was the old system type 4. A type D pathology will be associated with mutation in the VCP gene. So once the brain has been given a diagnosis by the neuropathologist, it then can be released for research at the brain bank and with our collaborators. And we also work with various research groups within UCL, nationally and internationally. We have a system in place at the brain bank where people can request tissue from us, so they fill in a form that tells us what research ideas they want to do. It's then passed to a committee and they'll decide whether we can release tissue for other research projects, both in the UK and internationally. The types of projects that the brain tissue is used for are obviously pathology projects, clinical pathological studies, the frozen tissue will be used in genetic studies, and biochemical investigations including different proteomic studies. So I just thought I'd highlight some of the studies that I've been involved with and the tissue's been used that's been donated to Queen Square Brain Bank. So this first study was a clinical path and biochemical investigation of a group of FTL cases diagnosed as FTLD FOS. So in their inclusions they have a protein deposited called fused-in sarcoma. So these are the types of inclusions that we see in the fused-in sarcoma. So we see the cytoplasmic inclusions similar to what we've seen in the tau diseases or the TDP diseases, but this time the protein's made up of the deposit's made up of the FUS protein. And this was a large study looking at 14 cases, it's quite a rare disease, looking at 14 cases and characterising the pathology of the disease now that the protein had been identified, and looking at the clinical manifestations of these cases. We then went on to look at these cases and probe them for another protein called transportin. This protein's involved in shuttling the FUS protein between different cellular compartments, and we found this protein was also in the inclusions, and this study led on to a PhD student starting at the brain bank to investigate the role of this protein in the disease. As I've said before, lots of the cases are involved in large clinical path studies. So this was a study that we did with John Rohrer at the Dementia Research Centre, looking at 95 cases. Many of the cases were donated prior to the current classification, neuropathological classification of FTD. So we had to go back to the cases, recut them, restain them with all the new antibodies, now all the proteins have been identified, and give them a formal neuropathological classification. Once this was done, we could then compare the clinical notes with what we'd found in the pathology. And we found some correlations, as I've just shown you in the table, that if somebody comes into clinic with a semantic dementia, and it's caused by a TDP pathology, then we can see that it will always be a subtype C. A behavioural variant, FTD, is still quite a mystery. If you come into clinic with FTD, you can have a tau pathology, TDP43 pathology, or a FUS pathology. So obviously more clinical path studies need to be done for us to be able to refine this even further. One interesting piece of work that came out from looking at the brains down the microscope was this paper that we published last year. and we found a brain that had a known progranulin mutation, also had a C9orf expansion repeat. And it was only by looking at the brain tissue that we picked this up, because we saw the characteristic P62 inclusions, and then this brain was then sent to the geneticist to confirm that this had the mutation. So it has both TDP43 pathology, shown here, and the characteristic pathology that we'd see with the C9orf72. And I just thought I'd briefly mention some of the research that I'm currently doing on my fellowship, and this is to investigate both pathologically and biochemically what proteins are involved in the mislocalization of TDP and FUS. So I've explained a few of the roles of these proteins. So both the proteins are found within the nucleus of the brain cells. So we have TDP here and FUS here. And we believe that they're shuttled, they bind to the RNA and are shuttled from the nucleus out into the cytoplasm of the cell. But to do this, they need to bind to other proteins, and the focus of my research fellowship is to look at a group of proteins called the HNRMP proteins, which are able to shuttle both FUS and TDP from the nucleus to the cytoplasm. In the previous study, I've already shown that we found transportin, which shuttles FUS from the cytoplasm to the nucleus, is found in the inclusions, implicating that this mechanism here might be disrupted, but we also want to investigate whether this mechanism here is disrupted within the brain cells. So this is an ongoing project, but briefly I just thought I'd show you that we found multiple proteins of the HNRMP proteins. It's a big superfamily of proteins. There's about 20 proteins in this superfamily, and I found 5 or 6 of the proteins to be in the cytoplasmic inclusions here, and also threads are present here. So the research that I'm doing now is to sample the frozen halves of the brain and biochemically see whether there's increase or decrease in these brains of these proteins that might lead us to more of a clue of what the mechanism involved in the disease is. So thank you, and any questions?